So I'm Jiacheng. I'm currently a postdoc at Stanford. But today I'm going to talk about some of my work that is kind of the last pieces of my PhD thesis, which I personally feel very excited about. So I will go through this topic. It's called Polygenic prediction of treatment efficacy with causal transfer learning. So what started with the motivation is that if you look at how many clinical trials that can started from like the phase one to the final approved by the FDA, you will see that 90% of trials actually fails. And here is a figure that shows what's the major failure reason of these clinical trials. And the purple color here is actually represent the efficacy, meaning that many of the drugs fails due to it fails to reach the predetermined efficacy that's required for the FDA to approve. So lack of efficacy is the primary reason for drug discovery, drug development failure. So the key questions we are interested in asking is, can we identify actually a subgroup of patients for whom actually the drug is effective so that the doctor can prescribe those drugs to these people who actually we will make sure this drug is beneficial for them. So that's the main goal. And so if we, and to frame it formally is typically called heterogeneous treatment effect. That is, we have some treatment, but it is only effective for individuals with specific characteristics. So here's one paper from JAMA like 2 years ago, and their treatment is like some therapeutic dose heparin for like what to see whether it can improve like the patient status for the like the COVID-19 patient status. And the result is that they found that actually this drug was more likely to be beneficial in patients with who were less severely ill at presentations or who have lower body mass index. So these are typically considered some sort of heterogeneous treatment effect that we may only prescribe those drugs to the patient who have these specific characteristics. So actually, and this is actually a very hot topic across multiple disciplines. So for example, in medicine, people are really curious about this heterogeneous treatment effect. And also because I think in genetics, people are also very curious about whether people typically call it gene involvement interactions is to see whether like the genetics will modify the policy effects or some treatment effects. And from like the method people, that is from the causal inference or from the machine learning folks, they also develop a lot of statistical and machine learning method, AI method, to help us to understand and identify this heterogeneous treatment effect. So here I will give you one example about one study that we have in hand. It's called the Lung Health Study. So it's a randomized clinical trial, but at 30 years ago. But it's I think this is a very interesting trial. So the treatment group is that they have the smoking interventions or some inhaler that with some active drug prescriptions. And the control group is that they want to understand whether they want to have like smoking interventions plus the placebo, but do not have these actual drugs. And their major question of interest is that for the smokers, if people have prescribed those drugs to this patient, because smokers, they will have like lung damage, and they want to understand whether for people who have these drugs will have a slow decline of their lung function, which is typically measured by the FEV1 levels over a 5-year period. So this is the number of people who participate in these studies and also have genetic variables. So in total, there are around 2,500 folks, and half of them are treated, that is randomized, and half of them is controlled. But if you look at the cumulative change of the FEV1, so you can imagine that suppose people who have a lot of like decrease of their FEV1 values, it means that their lung function is damaged a lot. So if this drug is effective, then you would imagine that suppose people who have this drug treated should have a slower or less decrease of this FEV1. But if you actually look at these studies, actually is people who are treated actually have more lung function like damaged compared with the control group, which is quite interesting. So actually, but this is not statistical significant. So the conclusion of the original study is that use of this bronchodilator does not influence the long-term decline of the FEV1. So this is very interesting data. And so I want to keep this data just throughout this talk, but I will mention what's the key limitations for the current heterogeneous treatment estimation method to work. So there are three papers that show you a lot of method that is saying that suppose you have a rich set of covariates, rich set of like patients characteristics measured, you can actually apply this method to this data and it can give you pretty good estimation of this heterogeneous treatment effect. So if we want to really estimate the heterogeneous treatment effect, it typically requires large sample size with like very rich pre-treatment feature set because we want the pre-treatment because we don't want to let anything to be influenced by the treatment that can capture the relevant variations among these individuals. So this is to our understanding the key limitation of the current that if we want to apply this method, this is the key limitations. And if you look at the long-term study, it only has 40 pretreatment features. And it is very, I think it's that people will typically measure something that they care about their studies. So it has the like the biological sex, age, and a lot of like baseline variables about their lung functions and about their BMI. So this is the things that typically measured, but it only has 40 features. And if you apply the state-of-the-art method on these 40 features to estimate the heterogeneous treatment effect labeled here, you can see here's the p-values. So there's actually no evidence saying that there's any evidence for the heterogeneous treatment effect. So this is what the typical pipeline look like. So you will get these features, and you will apply the best method, but you will typically get no results. This is the question we aim to address. So we are thinking that maybe the personalized genetic medicine is the future for healthcare. That is based on people's DNA variations, we can decide whether to prescribe some drugs to the people or not, because we are able to predict based on their DNA whether this drug will be effective for them. And I will try to convince you that this is at the frontier and the things we should pursue in the future studies of these clinical studies. So I will also use this example. So I've mentioned this is a study that try to understand whether use of these drugs will influence the long-term decline of the FEV1. And the conclusion of the original study is no. And if you apply the current best HTE estimation method, the answer is also like there's no heterogeneous treatment effect. But here is the method. I will introduce the details later, but I will show you some examples. That is, if you apply our method on using the genetic variations of these patients, you will typically get a very significant heterogeneous treatment effect estimations, in contrast to the current method that people use in genetics field, or the same method but using non-genetic features. So this is saying that we are trying to develop method that is can handle these very high-dimensional genetic variations, and also it can help us to really identify the heterogeneous treatment effect. And if we actually want to predict the individuals based on our predicted efficacy, as you can see here, we predict that those people will have this drug will do harm for them, and we predict that for these people, the drug will do good to them. And actually, you could see a pretty like monotone trend saying that our prediction is pretty good. That's saying that we can stratify the patients into different groups and predict whether the treatment is effective for them or not. And then the doctor can decide whether to prescribe the drugs for them or not. So this is like, I will first show some results, but later go into the details of the method. So here's the method part. So we try to formalize what we are talking about when we talk about using genetic variations to predict the treatment efficacy. So I will introduce very little math here, try to help you understand the question. So this is the model we call the polygenic GY treatment models. So this is the outcome of the clinical trials. and we have the genetic main effects, additive effect that will contribute to the main outcome, and treatment effects contribute to the outcome of clinical trials. But we will also have this G by treatment interactions, saying that actually the treatment effects actually depends on the people's genetic variations that will lead to different response to the same drug. And so we define a key quantity here called polygenic efficacy score. That is the weighted sum of all the SNP variations across the genome. But the weight here is come from this interaction effect rather than the main effect. So if you're familiar with the PRS literatures, people typically consider using this weight as the PRS. But we define the polygenic efficacy score using, for example, using the interaction effect. And we show actually a pretty interesting equations here is that the KATE, that is the typical quantity that people are interested in estimating heterogeneous treatment effect, called conditional average treatment effect, is actually a linear function of the polygenic efficacy score we define, saying that people who have higher scores, the treatment will do good to them. So this is some math trying to show you the intuitions and why our definition of the polygenic efficacy score makes sense. But it's actually quite challenging to actually estimate these parameters or estimate these scores. Because if we have the oracle like the beta ij here, it will be extremely easy to estimate this quantity. But typically we get very noisy estimates due to the small sample size of the randomized clinical trials. So what we are proposing here is we try to borrow the success of these current language models, saying that we should not train the model from scratch. Instead, we should fine-tune the existing models in some small data. But the fine-tuning should also give you very good performance. So we have the similar analog here is that instead of trying to estimate how each SNP, how each genetic variations contributes to the treatment effect efficacy, we first using the pre-trained model, that is the pre-trained polygenic risk scores, and to calculate the scores, but we are using the randomized neural trials to fine-tune these scores. And you can imagine this as some dimensional reductions, and then to give you a final polygenic efficacy score. So this is not training from scratch, but just fine-tune these well-powered PRS models in some randomized clinical trials. And we will show that this is actually can help you a lot in estimating these polygenic efficacy things. And so this is the whole pipeline of our method. The idea is that the input is you need to have the individuals with the treatment response, treatment assignment, whether the individual receives treatment, but also their genome-wide genetic variations. But instead of training the model from this matrix, you can consider it as some feature matrix. We were first using some pre-trained genetic risk prediction models to do dimension reductions from this tens of millions of SNPs into thousands of PRS. And then we will apply this causal machine learning that is trying to fine-tune this matrix to estimate the polygenic efficacy scores. And we will do some sequential inference to make sure that the training fault and the inference fault is not the same fault, so there's no overfitting problem. And finally, the output of our method contains the p-values to see whether genetic variations contributes to the heterogeneous treatment effect. What's the top PRS the feature interpretations contributes to the polygenic efficacies. And we are able to predict whether individuals were more likely to respond to the treatment or not. So this is the whole pipeline of our method. But you can imagine that it is actually quite hard to get this randomized clinical trial data. So we also developed one method that is using the summary stats. That is, we only requires the G by treatment summary stats to help you do the inference. So we have the individual level data at mLearner, and we also have the summary stats based on mLearner. So next, I will show some, I think this is, to me, pretty cool results about the power of this fine-tuning instead of training the model from scratch. So here I'm using the power, that is the power to detect, statistical power to detect this heterogeneous treatment effect. And the panel here shows training model from scratch. So the x-axis is the proportion of the treatment variance explained by the G by treatment interactions. Y-axis is the power. So as you can see, this is the realistic range is 0% to 5%. This is actually pretty large effects. But if you training the model from scratch here, you can get almost no power, like at less than 10% of power to get any meaningful results, meaning that you cannot have like various different p-values to convince you that the genetic variations will help us a lot in like explaining heterogeneous gene effect. But if you are using the transfer learning, as you can see here, it actually can give you pretty good estimations. This is the typical range. So we define a thing called transferability here is the correlations between the true and estimated PES. And this is the realistic range. As you can see here, we can get a huge boost of power compared with the training model from scratch. So this is because this PRS model is pretty good. It's all you train using this large scale GWAS data, which contains like hundreds of thousands of individuals, which can give you pretty good power. And we also do some simulations compared with alternative methods. And these two settings is about the type 1 error, whether we can whether there are any false positives. And one thing to note that is our method cannot handle the linear setting. That is the formula I showed before, but we can also handle nonlinear settings. And here's the power plot. As you can see here, like with very tiny randomized control samples, our method is still to give you pretty good powers, but all these methods cannot give you like any similar results due to this small sample size of the randomized clinical trials. And this is we want to show that actually the mLearner with summary stats can also help you identify the heterogeneous treatment effects. So this is a paper published in AGHG 2 years ago, and they have a very good clinical trial data, but they only release the summary stats, which is understandable because they are pharma, so pharma typically do not release the randomized clinical trial data. But their results is that this treatment is actually equally effective regardless of individuals' genetic backgrounds. So this is the major findings that are now results for their paper. But if you apply mLearner to the same data, you can actually get pretty significant results. And for the ankylosing spondylitis, we found that mLearner can give you very strong evidence saying that that there's very huge genetic explained heterogeneous treatment effect. So the x-axis here is different outcome that measured in the clinical trials. Y-axis is the minus log 10 p-values, and this is the p-value equals to 5%. And our method can give you pretty significant results across like the primary outcome for the ankylosing spondylitis, but also the secondary outcome, like for the biomarker for the ankylosing spondylitis. And we actually found that the PRS for the bone mineral density is the top predictors if we range by like feature importance. And this is consistent with like three different outcomes that is related to the ankylosing spondylitis. And also we try to use that this is what people typically do, just use the outcome PRS and train the models, and it does not give you any simple results. So only the PRS we identified will give you pretty simple results. So the other case study I have already mentioned is the long haul study, which we also show that mLearner with individual data also can give you pretty good results. So this is the case study we have in our paper. And software is ready to use. It's actually very easy to use. It's available on GitHub right now. It's called mLearner, and you only need to have the outcome treatment and PRS matrix, and then our software will handle those for you. It's pretty fast, like less than 10 minutes, you will get the results. So to summarize, we developed mLearner as a transfer learning and genetic powered estimation of the heterogeneous treatment effect. And we have two case studies. One shows that low mineral densities as a key determinant of this drug's efficacy in anti-IL-17 drugs efficacy in ankylosing spondylitis. And we also identify a small subpopulation adversely affected by a bronchodilator treatment. And we actually, I think important contribution is that we show the utility of using genetic variations in the inference for the heterogeneous treatment effects. So I want to talk about what we are thinking is the future for HT estimations with genetic variations. So the first thing is that we should fine-tune existing models instead of training from scratch. So I think that's some sort of the bitter lessons that people have already learned in this AI field, but I think genetics people should also adopt this analog here. And also that the pharma or the people should release actually the randomized control data. And we also show that summary level data should also work. The third thing is that genetic variations actually matters for the efficacy of some drugs. So please do genotyping array or sequencing for randomized control populations. And we also, I think the final thing is quite interesting because actually, if you think about that, the actual, although like the biomarkers that explains the efficacy may not be the genetic variations, but we consider genetic variations as a universal causal instrument for the true biomarkers you typically identify. For example, the bone mineral density can be actually the causal biomarkers, but people do not measure that in the clinical trial cohort. But with genetic as instrument, people can measure that retrospectively. So I want to thank to my all co-authors, especially my PhD advisor, Lauren and Chongshi, who helps me a lot for this study.